Our next group of Coleopterous borers are the bark and ambrosia beetles. As stated in the introduction to borers module, these beetles are now in the weevil family Curculionidae and in the same subfamily, the Scolytinae, but they have very different feeding behaviors. Bark beetles stay in the sapwood of the tree and eat the tree fibers, but the ambrosia beetles bore deep into the xylem to make brood chambers that are infested with a fungus that is eaten by the larvae. For a long time, bark beetles and ambrosia beetles were in their own family, the Scolytinae. However, genetic analysis has now shown that they are really weevils, but weevils without snouts. So now they are in a subfamily of weevils. The head is largely hidden by an overlapping of the pronotum. Both beetles are generally quite small, usually being less than 1/8 of an inch in diameter and a 1/4 inch long or less. There are some larger species, but these are uncommon in landscapes. Bark beetles are also called Engraver beetles because their larvae make shallow burrows in the phloem and cambium zones. When the bark is removed, the undersurface of the bark and the surface of the tree trunk can look like something engraved the surface. The beetles enter trees by making small, very round holes, as do the new beetles that emerge. These are about the size of buckshot. So, shothole beetles is another common name. There are a few bark beetles that attack smaller branches or twigs, and a few also bore into shoots. The vast majority of these beetles are opportunists that quickly attack trees and shrubs that have a compromised vascular system, either from drought, disease, or physical damage. Some are also implicated in transmission of fungal and bacterial diseases. I'm going to use this image of completed hickory bark beetle burrows. This is a rather classic bark beetle in its biology and larval behavior. The adults of this beetle colonize recently killed hickory trees or trees and branches that are dying from vascular disease. Notice that there are 2 brood engravings in this image. When a bark beetle female finds a tree that doesn't cast her out with sap, she burrows through the bark to the phloem and cambium zone. Here, she makes a more or less linear burrow or gallery in this zone. In the case of the hickory bark beetle, the female makes her initial burrow in the direction of the wood grain. Other species make burrows across the grain, and the direction of the burrows can help in identification of which bark beetle species you are dealing with. The initial burrow is called the nuptial chamber, egg chamber, or egg gallery. This is because many of the bark beetles don't mate until this initial chamber is constructed. Some species have males that remain in the nuptial chamber, and in other species only the female remains. Soon the female chews small niches along the edge of the nuptial chamber where she inserts a single egg in each niche. Notice that as the larvae burrow away from the nuptial chamber, they increase in size, which also increases the width of their burrows. In the case of the hickory bark beetle, the larvae take 6 to 8 weeks to complete their larval development. The larvae are grub-like and they lack legs. After finishing their development, the larvae enlarge the burrow a bit and often point the burrow into the lower bark. The pupae are formed in these chambers. It often takes several weeks for the pupa to finish its development, and the new beetles can be easily seen through the thin pupal exoskeletons. In northern zones, this beetle has 1 generation with mature larvae overwintering in their pupal chambers. Pupation occurs in early spring, and the adults emerge from May to August. These feed on hickory buds and bark for about 2 weeks before they are ready to make their own nuptial chambers. In southern regions, the adults emerge in April through June, lay eggs in complete development of a second generation by August, and those adults make a second set of nuptial chambers and have a second brood. One of the most spectacular insect and plant disease stories is that of the Dutch elm disease. The disease is believed to have actually come from Asia, but because some Dutch plant pathologists were working on the disease in the 1920s, it got its name from that research. In the 1930s, the disease was accidentally introduced into North America, and since that time, it has devastated the large American elms that were used extensively as street and landscape trees across North America. The fungal disease slowly clogs the vascular system of the elm trees, which can kill the trees in 1 to 3 years after initial infection. The bark beetles that invade infected trees are merged with the fungus contaminating their bodies. When the beetles do some maturation feeding on elm bark and buds, they infect the tree with the fungus. The fungus can also spread through root grafts where the roots of 2 trees join underground. The native elm bark beetle can transmit this disease, but 2 non-native bark beetles are much more efficient vectors. The smaller European elm bark beetle is an invasive species that is common in eastern North America. It is aggressive in its attacks and is a very efficient transmitter of Dutch elm disease. The native elm bark beetle seems to be less aggressive and generally attacks elm branches that have been damaged or elm trees that are dying. In the western states, the banded elm bark beetle is another aggressive and efficient vector. Remember that all these bark beetles accidentally transmit the fungus through their maturation feeding. Upon emerging, the new adults immediately nibble on the bark of small branches, often in a fork of the branch. Bark beetle larvae are small, grub-like, but lack legs like the rest of their weevil cousins. Let's take a minute to review the interrelationship of the 3 elm bark beetles and Dutch elm disease. While the beetles and the disease can operate independently, they both have a synergistic influence on each other. The emerging beetles are unwitting carriers of the fungal spores. When the adults do maturation feeding on small elm branches and leaf buds, the spores can invade the damaged tissues. The beetles prefer healthy trees for their maturation feeding, so the fungus gains entry into those healthy trees. When ready to make their nuptial chambers, the beetles prefer weak, dying, or recently killed trees. In short, a year after the beetles infected a healthy tree, the infected tree is usually weak and in the process of dying, which is highly attractive to the beetles of that year. Beetles using these trees are also the ones most exposed to the Dutch elm disease fungus. So, bark beetles vector the fungus, which creates stressed trees that the beetles can effectively utilize in the future. Another aggressive bark beetle is the Shothole Borer. This pest is also known as the fruit tree bark beetle. It commonly attacks cherry, plum, peach, and apple trees. The adult beetles are active for much of the summer, and they readily attack these host trees, especially when the trees are under vascular stress. The vascular stress can be transient, as in drought conditions, or because a disease or other borers have girdled the tree. When it attacks weak trees, gummy resin can be produced by the tree, but mass attacks by the beetle can overcome these defenses. This pest can have 2 generations in southern zones, but only 1 generation in northern regions. It overwinters as nearly mature larvae that finish development in the next spring, pupate, and emerge by late May into June. I get inquiries about this borer from owners of purple leaf flowering plum trees. The claims are that the tree just suddenly died and the shot holes appeared in the bark. In most of these cases, peace tree borers, one of the clearwing moths, had girdled the tree below the mulch, and the shot hole borers just took advantage of the dying tree. There are dozens of bark beetles that attack conifers. In pines, the genus Dendroctonus includes the southern, western, and mountain pine beetles, plus the black and red turpentine beetles. These are major pine forest pests that are able to perform mass attacks, especially when forest trees are under drought stress. These beetles are some of the larger species, and when they attack fairly healthy trees, large pitch masses can form where the beetles are entering the trees. The spruce beetle is a dendroctonous species that specializes on spruces. Dendroctonous females make the nuptial chambers, then release a pheromone to attract males for mating. 2 or 3 generations in a season can occur. Ips beetles are smaller species and are more attracted to recently killed or dying pines and spruces. Most Ips species overwinter as adults in pupal chambers. They emerge in early spring and search for dead or dying trees. Males construct the nuptial chamber, then release a pheromone to attract a female. After mating, the female chews egg niches and deposits eggs. Because the host trees are dying or have recently died, these beetles expel dry sawdust-like material from their galleries. The larvae burrow laterally in the sapwood area and take about a month to develop. There can be 2 to 4 generations per year of Ips beetles. The eastern juniper bark beetle commonly attacks eastern red cedar, but it also attacks northern white cedar. In northwest Ohio, it has been found attacking agricultural windbreaks of white cedar that were planted side by side along roadways. Mature trees are often stressed by hot summer weather and drought, as well as winter road salt. Such stressed trees are colonized by the beetles, which make short nuptial chambers following the wood grain. The larvae move laterally a short distance and then also follow the wood grain in making their burrows. Unfortunately, this beetle often carries a wood rot fungus that can colonize trees through the nuptial chambers. In healthy trees, the bark beetles alone cause minimal damage, but with the fungus, trees can be killed. Before we get deep into the ambrosia beetles, let's quickly review the difference between bark beetles and ambrosia beetles. Bark beetles mainly utilize the phloem and cambium, possibly the outer layer of xylem. Ambrosia beetle females burrow directly into the xylem, often into deeper layers. When bark beetles attack, there often is pitch or sap flow or dry sawdust-like frass pushed out of the entrance hole. Ambrosia beetle females do not eat the wood but push out tightly packed wood dust that can emerge from the hole, which is often called a toothpick. Bark beetle females insert eggs into niches along the nuptial chamber, while ambrosia beetle females deposit the eggs loosely in their ambrosia chambers. Bark beetle larvae eat the wood, while ambrosia beetle larvae eat their cultivated fungus, the ambrosia. Bark beetles are specialists that attack heavily stressed or recently killed trees, but ambrosia beetles seem to be able to attack more healthy trees. New bark beetle adults chew their own exit holes, while the ambrosia beetle new adults emerge out of the parent's initial burrow opening. Like the bark beetles, the ambrosia beetles used to be in the family Scolytinae and some species in a related family, the Platypodinae. Both are now subfamilies of the Curculionidae, the weevils. Platypodid adults are elongate beetles with prominent antennae, while the Scolytid adults are very short with the head deflexed under the pronotum, and they often have truncated abdomens. Ambrosia beetle adults are rarely seen because of their small size, but once they start an attack, their presence is easily observed. The females do not eat the host tissues as they enter the plant. They simply chew small shavings which are pushed behind them. These shavings tend to stick together and begin to emerge out of the entrance hole. Since they look a bit like a toothpick stuck into the bark, they are commonly called toothpicks. These toothpicks are easily broken in wind or rainstorms, so they can be easily missed, especially in landscape trees. Ambrosia beetle females have various structures that are modified for carrying fungal mycelium with them. These structures are called mycangia. Depending on the beetle genus, the mycangia can be on the head, pronotum, or pits located in the exoskeleton that may be scattered over the beetle's body. Different ambrosia beetle females make variously shaped burrows into the xylem. Some are single burrows and others can have 2 or more branches. After the brood burrow has been cleaned out, the beetle removes the fungus spores from her mycangia and applies it to the burrow walls. The moist conditions allow for rapid fungal growth. Eggs are deposited, and upon hatching, the larvae feed on the fungal hyphae. After completing their development, the larvae pupate, then emerge as new beetles out of the brood burrow opening. Unfortunately, various bacteria and other fungi can contaminate the mycangial pouches, and these can be carried by the female beetle to new trees. The most common bacteria is one that causes staining of the heartwood. This is usually called blue stain, and it is a downgrade for lumber. While most ambrosia beetle attacks are to smaller diameter trees, those that have 3 to 5 inch caliper trunks, there are some ambrosia beetles that can infest smaller branches. These small branches can be weakened by the attacks that are still in the xylem, but may occupy more than half of the xylem space. When windstorms occur, these infested branches can break out. In other cases, the attacks to small branches can actually girdle the vascular system, and the branch will flag or suddenly wilt and die. Most of the native ambrosia beetles have been considered to be primarily nuisance pests in forest production. Their attacks and resulting blue-stained lumber diminish profits in lumbering operations. However, over a dozen species of Asian ambrosia beetles have been recently introduced, and these aggressively attack smaller trees. These have become major pests in nurseries that specialize in producing deciduous trees and shrubs. Attacks are often extensive, and damaged trees can't be sold. Small trees and landscapes also can get attacked, and while the attacks may not kill the trees, their growth will be greatly slowed. Unfortunately, nursery tree producers and homeowners rarely discover the attacks until after they have occurred. Since most systemic insecticides don't penetrate the xylem tissues, curative treatments are useless. Also, attacks often occur very early in the season when plant managers are not expecting pest activity. Two of the more important Asian ambrosia beetles are the Granulate ambrosia beetle and the black stem borer. The Granulate ambrosia beetle is more active in southern zones, and the black stem borer is an important pest of northern deciduous saplings. Both are quite small beetles, with the adults being just under 3 millimeters in length. Their burrows are only about 1 millimeter in diameter. Both overwinter as adults in old burrows or in leaf litter. In early spring, the black stem borer females fly to locate potential host trees. When a suitable tree is located, the female starts to burrow through the bark, phloem, and cambium, and well into the xylem, while she pushes out the wood in a conspicuous toothpick. Upon cleaning out her burrow, the female establishes her fungal ambrosia, lays eggs, and may depart and construct an additional burrow. There can be up to 2 generations per season. Recent research by USDA forest entomologists have confirmed that the black stem borer is the most common ambrosia beetle attacking small deciduous trees in Ohio nurseries. The adults seem to key in on trees that are waterlogged in the spring. Apparently, having waterlogged roots causes the plants to produce alcohol. This alcohol odor is a strong attractant, and beetles will first attempt to enter these trees. The overwintered females attack plants in the spring from early April into mid-May. It takes about 2 months to complete the larval and pupal development, which leads to a second generation of adults being active in July and August. Again, waterlogged trees are preferred in the summer time period. Since Ambrosia beetle adults seem to be highly attracted to ethyl alcohol odors, a simple trap has been developed for nursery producers to use. This trap consists of a 2-liter soft drink bottle attached to poles that are located in the nursery, hopefully near susceptible trees. Longitudinal slits are cut into the bottle and removed. A dental wicking is placed in a pill vial filled with ethyl alcohol. The ethyl alcohol can be easily obtained as shellac thinner at most hardware stores. This vial is wired to the interior of the trap. The trap is attached with its top facing down, and the bottom is now filled with detergent water or a dilute antifreeze solution. Both will capture any beetles that enter the trap. The little beetles are easily observed in the clear plastic bottles, and when first detected, bark-applied preventive insecticides can be sprayed onto susceptible trees. To summarize our current approach to managing attacks by Asian Ambrosia beetles, we now know that control attempts after the beetles have entered the trees are not effective. This is really problematic in landscapes because preventive treatments are usually not worth the economic investment. In nurseries, traps can be used to monitor adult beetle flights. As soon as adult beetles arrive in the traps, susceptible trees, mainly small deciduous trees that may be growing in wet soils, need to have their bark covered with insecticides known to be able to kill the adults trying to enter those trees. Some of the insecticides can only be used in nurseries, not in landscape trees. Because of the short residual action of these bark sprays, repeat applications may be needed, especially if beetles are still being trapped 10 to 14 days after the initial application. Infested nursery trees should be removed and chipped to kill developing larvae. Favored trees should also be grown in areas that are not prone to flooding in the spring or during summer rain events.